Oh, grand. Cool. Oh, yeah. Yeah, that's up there.
Is that working okay?
Oh, it's just disappeared.
Oh, sorry. And here we go.
Okay, do you see the display settings? You might just need to swap them up the top. Yeah, there you go.
Is it swapped, or is it duplicate?
No, it's perfect there now. It's working.
Oh, it's working fine. Oh, yeah.
Oh, yeah, working fine there.
No problem. We're good to kick off. Everyone back?
All
Yep.
good, yeah.
All good. Perfect. Hello, everybody.
Yeah.
My name is Padraig O'Neill, and I'm the senior microbiologist here at Boston Scientific Cork. I'm just going to be giving you an overview of the microbiology function today, what we do, why we do it, and then what impact you can have as well on the micro function. So that's just the team. So what we will be going through today is our control environments, contamination, microorganisms, bioburden, and endotoxin. And then if you can see that plate there on the right hand side, so that's a nice, what we would call a worst case scenario. So you can see loads of little bacterial colonies there in the yellow, and then we've yeast and mold there as well. So that's what we don't want to see when we're counting our plates. Okay, so just an overview of the control environment. So all our clean rooms, we call them control environments. So you'll see them labeled when you're on site, or C1, C2, etc. We call them controlled because we're controlling kind of most aspects about them. So environmental conditions, temperature, humidity, pressure, they're kept within strict limits. We also do micro testing there on a monthly basis to make sure that the contamination levels are staying within limits as well. So we know they're not completely aseptic, that there is zero microbes inside there, but we have to make sure that it's staying within a certain level to make sure we do that. So we also have a cleaning regime, so we'd say contract cleaning on the floors, doors, etc. And then we have manufacturing cleans then on all our work surfaces. So there in the picture you can see the manufacturing benches there up and down, and all those surfaces will be clean on a daily basis, on a shift basis, actually twice per shift with IPA, so isopropyl alcohol, and that's just making sure that no contamination allowed build up over time, and it just prevents anything then from transferring over onto the devices. At the top of the pictures there as well, you can see the vents. So that's all the filtered air coming into the room. So it comes in from the top, comes down then over everybody and everything in the room. And again, that's just make sure there's no contamination building up over time. And everything is then going down to floor level. And then there's return air ducts then all around the rooms that are drawing that back out to be filtered and recirculated into the room. So just what is contamination? So it's any unwanted substance in a system or product. So you'd have your non-viables contamination, so soil, dust, sand, fibers, your piece of packaging, piece of metal, anything that isn't supposed to be physically in there. And then we have our microbial contamination as well, which we will be looking for, which is bacteria, yeast, and mold. So sources of contamination. So a bit of an interaction with you. So does anybody want to take a guess what is the greatest source of contamination in the controlled environments?
Personnel.
Personnel. Anybody else want to have a stab at it?
Skin particles.
Skin particles, yeah. So that's exactly right. So people. So 80% of our contamination that we pick up in the room is from the people inside there. 15% equipment and 5% the room. So all those lovely people are always trying to contaminate the rooms, and we're doing our best to stop that. So you're now part of that group as well. So microorganisms. So a single cell organism that cannot be seen with the naked eye. They're found almost everywhere and they reproduce very rapidly. So when I was talking about the contamination control, inside the room, so all the cleaning, etc. That's because we know once it's in there that it is going to become large numbers very quickly. So that's why we have all those cleaning steps in place to stop that from happening, and air changes and the filtered air. We also have gowning control. So when you go into the room, you'll see when you go into the gowning room, you put on a gown over your street clothes, and that's again to prevent any contamination that's on you entering the room. Again, just a bit of interaction with you. Where would you say is the greatest source of microorganisms on a person?
Their hands.
Or hands.
Hands. Mouth. Anybody else?
Clothing.
Clothing. Very good.
Hair.
Any other guesses?
Feet.
Feet.
Hair.
Hair. Yeah, getting closer there now to hair. So the scalp. So up by the head. So 1 centimeter squared, you're looking at around 1 million microorganisms. Then 4 to 10,000, and then you're looking there. A hands then could be 100 to 1,000, so that's different, Joe, for everybody. And then when sitting quietly, people shed up to 100,000 particles per minute. So I know someone mentioned there about skin particles, so that's very much something that can happen in the rooms. Is the old people, even when they're sitting quietly, they're shedding particles up to 100,000. But when you're actually in the room and you're working and you're walking around and you're building the product, that obviously can increase. So it's by wearing the gowns and wearing the hairnet and beer covers, etc. That's again all limiting the contamination that's actually been shed to the room. Just something to watch out, and I suppose especially in the COVID environment, something that we're very conscious of is coughing and sneezing sprays millions of bacteria into the surrounding environment. It's a lovely picture of a sneeze in action. So if you cough, sneeze, cannot be avoided, cough or sneeze away from the product and cover your mouth and nose and immediately wash your hands. At the moment, we're very much mask wearing on site and in the clean rooms, so we have a bit more control now in regards to coughing and sneezing. One of our biggest contamination control steps as well to prevent our products from being contaminated is hand washing. So every time you enter the controlled environments, you have to wash your hands. So you can see on the left there, that's a handprint with someone before they wash their hands, and then on the right is after they wash their hands. And then every one of those then is a colony of bacteria that's after growing. So that was if someone hadn't washed their hands and then they went in and handled the product, that would all be transferring onto the devices. So it's just make sure that you wash your hands. It's about 20 seconds wash is the minimum with the special soap that we have as you go in. And again, you might be going in and out of the rooms on multiple times throughout the day, but you should wash your hands the same amount of time in the first times you're going in as the last time. There's no dispensation for going in and out multiple times. And then hand drying then is equally important. And that is due to endotoxin. So endotoxin is released from water-loving bacteria when they die. If endotoxin enters the bloodstream, it can be fatal. So your body reacts as if there's an infection after happening, and it can cause a fever response, and you can go into a respiratory shock. We can't remove it by sterilization. So all our products we make on site here, we send to be sterilized. But endotoxin, because it's not actually alive, it's only part of a bacteria, it can't be removed by sterilization. So to ensure then we control the levels that are allowed on the devices through our micro testing. So every product inside there, we take representative samples and test them to make sure that the endotoxin levels are remaining very low. And we also, because water is such a large source of endotoxin, we test the water sources as well. And again, that's why when you're drying your hands going in, it's very important to have very dry hands when you go into the rooms after your hand washing, so you're not bringing in another risk of endotoxin for the product. So just to go over the risk contaminations again. So even though we do sterilize our products, there's always a risk of contamination. The sterilization cycles have limits based on our expectations. So we can't expect if we send millions and millions and millions of bacteria into the sterilizer that we're going to get sterile product at the end. So that's why we have to make sure through all our controls in place that we're limiting what's actually onto devices. And then as a reminder that endotoxin hairs cannot be removed by sterilization. And when you handle the products, patient safety is in your hands. I suppose that's something that can be forgotten sometimes, Joe, if you're concentrating on your numbers, etc., and what you need to be done. But every one of these products is going to be used on someone and as a patient. And last thing there, so we have our bullseye of patient care, and then quality compliance is where the microbiology area sits. Does anybody have any questions on the microbiology function?
I just had a question on your controlled environments. Are all of your clean rooms like ISO 8?
Correct, yeah, they're all ISO 8, so they're all validated to the same ISO, and they're all tested into the same micro limits.
Okay, awesome. Thank you.
No problem at all. Anybody else have any other questions?
Take the silence as a good thing, project.
Science is always good. Silence is golden. Well, thank you very much, guys. And I'm sure if you have any other questions, you can follow up with me after. There's no problem.
Thank you.
Thank you.
Thanks.
No problem at all.
Thank you. Thank you.
Okay, guys, so that is